analysis, right? We present that. So now I'm going to bring a journal in front of us that I think was a little controversial. So this is a journal in relation to melatonin causing heart failure. So this is actually a 2025 study. But again, this will be in headlines. If you haven't heard us, I've talked to A4M colleagues about this. My students and even the patients here are just going crazy. Should I stop my melatonin? Because it's related to heart failure risk. So let's slow this down and redefine print first and do what physicians do best and contextualize data. So this is actually an analysis coming from a large preliminary observational study presented at the American Heart Association Scientific Sessions in 2025 using electronic medical record data across US, UK, and other countries involving 130,000 adults with diagnosed chronic insomnia, of course, using the identifier, the ICD code of F51. Key point here that matters, when you include the UK, in the UK, melatonin is actually a prescription-only drug. So it's a drug. So melatonin use here is not casual supplement use in this study. It likely reflects more severe insomnia, physician involvement, and higher baseline medical complexity. So this is already a very different population than the average wellness patient that we see taking 0.5 milligrams at night that we use to, again, I guess, handle in our practices. So the reported outcomes here, so let me tell you the studies result. Over 5 years of follow-up, incidental heart failure for melatonin users, 5%. Non-users, 2 to 3%, so leading to a hazard ratio of 1.8 to 1.9. What does that mean? 90% higher relative risk when you're using melatonin for an incidental heart failure. Another one would be heart failure hospitalizations. So 19% for melatonin users versus 6 to 7% for non-users. So 3.5-fold relative risk. So all-cause mortality, 7.8 for the users and 4.3 for the non-users. Thus, twofold higher risk. So important reminders here, these are relative risks, not absolute proofs of causation. So here are several critical limitations or when we kind of criticize our journals. So here in the study, the confounding by indication, meaning chronic insomnia itself is strongly associated usually with sympathetic overdrive, hypertension, inflammation, and cardiometabolic disease. Thus, melatonin here likely marks sicker sleepers, not a toxic exposure. Number two, reverse causation. Early subclinical heart failure causes nocturnal dyspnea, fragmented sleep, circadian disruption. So patients may take melatonin because their heart failure is already developing. And number three, incomplete transparency. This work is currently an abstract, a press release, and some media summaries are included in this when you look at it in the internet. And again, full methodology, model specifications, dose stratifications, and sensitivity analysis have not yet been publicly vetted. So this is where balance matters. So multiple small clinical trials in patients with established heart failure shows, so improve NYHA classifications. So this is Guido et al. 2003, showed stable chronic heart failure benefited from melatonin using 5 to 10 milligrams and improved quality of life from Brugger P et al. 2011. And even when you use melatonin, it trends towards improved left ventricle ejection fraction. So mechanistic literature here is you can use melatonin in heart failure and even ischemic reperfusion injury. So consistency, it has been showing melatonin as a good antioxidant, anti-inflammatory, autonomic stabilizer, and mitochondrial protector. That biological points or characteristics of melatonin, it actually leads us towards protection and not harm. So the most accurate interpretation here is not melatonin causes heart failure, but it is possibly chronic insomnia is dangerous. Melatonin identifies a higher risk phenotype, and long-term nightly use should prompt cardiovascular and sleep depth evaluation. So melatonin is likely a risk marker, not a risk driver in this specific study. So thank you. And no, you don't need to ban melatonin from your practice based on a press release. And that's our tip of the week.
Yeah. So Dr. Leonard says firemen are often at the scene of a fire. Are they the cause of the fire? Well, most of the time not. But back home in Wilkes-Barre, PA, there was actually a scandal where firefighters were setting fires and then rushing, reporting them rushing in to save the day. So it was probably about 15, 20 years ago that they were actually setting fires themselves.
Well, yeah, remember this is coming from American Heart Association, right?
Right.
So this is not our, I guess what, our
Right.
base, our
Right.
foundation.
That's not our base, no.
Yeah. So,
You know.
but just to give our clinicians heads up here,
Mhm.
don't be flustered by this data. We got questioned about this in AFRM when we were doing a lecture, and some of us haven't read the literature yet. So I did a deep dive about this to kind of tell you that this is really not right now. It's not true yet.
Okay. Dr. Andrews, is this your slide here? Why is cancer has not been cured or?
Oh yeah, you can see that already? Okay,
Yeah,
good.
yeah, yeah. Okay. I thought it was Dr. Narios. Okay. So I was going to ask him the question, but now we'll get to you in a second here.
The
Okay.
million-dollar question.
Somebody asked, we were talking about battlefield acupuncture. Yeah, the ASP needles, that's asked, do I use, yeah, those are the ones I use when the patient to go home. And Dr. Nario today, so we use them in the office too, in the clinic, and I'll use body needles there in the ear for that. And then take them out and then put the semi-permanent ones to go home. It's much better out in the West Coast. It depends on actually on where you are. When I first learned how to do this, I was in Pennsylvania where there's a lot of humidity, and the needles would come out. The home needles they would come out in a day or two. Out here, they'll last anywhere from 3 days to a week, and they come out on their own.
Yeah, so well, if you come here to our clinic, you'll see all my staff with needles on their ears. Unfortunately, they're my lab rats
Yeah, okay.
because we're going to be tested in school. So,
You're gonna be.
so when you come here to our clinic, it's all good. They're safe. I got their consents.
And Dr. Leonard, everything happens in Wilkes-Barre. So actually, the saying is everything happens to me and Dick Tracy in Wilkes-Barre. So if you remember, there was a cartoon called Dick Tracy. It was it was
Yeah, with the hat,
a
the guy
cartoon.
with the hat.
Guy,
Watch.
his name was Al Cap, and he was actually from Wilkes-Barre. He was the guy who drew it. And there's a statue of him actually going from downtown Wilkes-Barre to a town called Mountaintop, about halfway up.
I say you became
That
famous there.
that's it. If somebody says to you, everything happens to me and Dick Tracy, you know they're from Northeast Pennsylvania. So among other things, they'll also say things like, I'm going to go up the mall, not to the mall, or down the shore. You know that from there. But be that as it may. Um, so tonight we're pleased to have Dr. Bill Andrews with us. And we were going to talk about the what, why, how, and how not to age. He's a
Okay.
fresh from a starring role at our recent Congress of Medical Excellence 5.0. Dr. Andrews is a molecular biologist, a biogerontologist. He's best known as the founder and president and CEO of Sierra Sciences, a company dedicated to extending human lifespan and health span through telomere research. Dr. Bill earned his PhD in molecular and population genetics from the University of Georgia in 1981. He's held key research roles at several biotech firms, including Gerion Corporation, where he led the team that identified RNA and protein components of the human telomerase. He's a principal discoverer of telomerase, a key enzyme in aging, and was awarded second place as National Inventor of the Year for his cancer research. He has over 50 patents and authored numerous scientific papers in the field of aging and telomere biology. So, Dr. Andrews, it's an honor to have you, and take the floor.
All right, let me bring my PowerPoint up.
Your mother must be proud.
Whose mother?
Yours, with a resume like that?
Oh, she was. It was, hold on, let me get how do I get I can't see my own screen because I've got a window. Okay, so good. Let me
It
just
says because.
all right, I have to, okay, good. So I want to begin with a pitch about something, a serious problem that I think needs to be resolved. And that has to do with why cancer hasn't been cured yet. And oops. Okay, so there it goes. Okay, so likewise, why hasn't heart disease and dementia and multiple sclerosis, muscular dystrophy, and aging, et cetera? Why haven't those been cured yet? And it's not because scientists don't know what to do. And that's what I want to discuss here. Okay, it's because in academic research, there is a problem of publish or perish. Is publish or perish, you publish something, you get a grant. Grants, you do more publish or perish, more publications so that you can get more grants, on and on and on and on. So the problem in academic research is the thing that people gauge their success on is by the number of publications they have, not on curing diseases. In commercial research, you start a company to do some research, you get an investor, suddenly you're not in control of that company. You don't own the company anymore. The investor owns the company. The investor wants a quick return on investment. Then the investor wants an exit strategy. Then he invests in another company, wants a quick return on investment, exit strategy, on and on and on. The gauge of success here, again, is not curing any diseases. It's how much money the investor makes. Then there's the companies that have been started by these celebrity billionaires. Okay, so they've hired celebrity officers. Those officers have hired celebrity advisors. Those advisors have hired celebrity scientists. And again, the gauge of success now is celebrity. It's being celebrities. That's what it's all is. What the problem is that there's no cures. Nothing is being cured because none of these things are working. The system's not working. It's broken. Okay, so the solution is that we need to get hold of very discrete, ultra high net worth individuals, not the well-known ultra high net worth individuals, because they're still, even though they have a lot of money, they're still focused on how much money they can make. And that's, in my opinion, a real problem because it doesn't lead to cures. Okay, the problem with this very discrete ultra high net worth individuals is just by their definition of being very discrete, how do we reach them? Well, the number one thing that all ultra high net worth individuals have to do is see their doctors. That's probably the one best conduit to get to these very discrete ultra high net worth individuals. So I'm calling for a call to action that I'm asking that doctors, who have patients that are considered very discrete ultra high net worth individuals, they should be telling these doctors that the diseases that even these individuals are suffering from, cancer, heart diseases, et cetera, and their loved ones are suffering from, they're not going to get cured unless they actually start doing something about it to help and at least help financially. And that's something they don't know. They believe that everybody else has things under control, and they need to know that they're not under control. And I have attached a document to the chat that I would like to have doctors provide to any of their patients that are considered ultra high net worth individuals to bring a solution to this kind of problem. All right. And that's just my pitch there at the beginning. I'm actually doing that at all my presentations now. So I've been obsessed with the subject of aging my entire life because I want to do something about it. All through school and stuff like that, I've been focused on trying to deal with how to understand aging really well. Okay, so I got a bachelor's degree in biology and experimental psychology. And the only reason I got a bachelor's degree in experimental psychology, besides the fact that psychology plays a big role in aging, is because in the bachelor's program, nobody was teaching you how to be a scientist. And so when I went to the counselors at the school at UC San Diego to complain about this, they said, well, go to experimental psychology. They teach you how to be a scientist. So I ended up learning a lot of different things, which I'm not going to go through, but just mathematical modeling, statistical theory, most things that most biologists don't know a thing about. Okay, key ones like construct validity on the bottom right or middle right, things like that. It's just unheard of in the biology field, but not in experimental psychology. I then went and got my PhD in population genetics and molecular genetics because, again, I wanted to learn what I needed to know about aging. Okay, so population genetics is the subject of how and why of evolution as opposed to the what and when. So I needed to know, for instance, why did we never evolve a way not to age? If aging was such a bad thing for us, why haven't we evolved a way not to age? So molecular genetics is how do genes work? And this pretty much tells us how we can solve all the problems. How can we succeed where evolution failed us? How can we fix the problems that exist now that evolution never solved for us? What I've learned as a result of all this is that from all this education and just obsession, putting all this arsenal of tools together, is that nobody knows what aging is. Nobody knows why we age. Nobody knows how we age, and nobody knows how not to age. This is something, even though there's a tremendous number of theories, a number of publications, books written on the subject, when you really start doing a lot of serious, deep thinking about it, like I have my entire life, you start realizing that everybody's got it wrong. It's all the twos and twos have to add up. There's no single theory that explains everything we know about aging. And so that's why I've been focused on that. And when I got my PhD, finally, I realized I still did not have enough information. I still needed to learn a little bit more before I could really start focusing on aging. And so I went into biotech instead, became a medical researcher in biotech. And I applied this arsenal of tools to the discovery of a lot of different things. I probably have more inventions, discoveries in the medical field to my credit than anybody else you've ever heard of. Okay, I'm one of the key inventors of human growth hormone, tissue plasminogen activator, erythropoietin, on and on and on. Lots of cancer drugs, including Rytelo, that's on the top, bottom right, that just got approved by the FDA last year. I've been the inventor or co-inventor of more biotech breakthroughs than anybody I've ever heard of. I was also awarded second place for National Inventor of the Year in the United States for my cancer research. And the key thing is that I am trying to focus on trying to find a cure for aging now, and I can cure aging, and my track record shows it. Okay, I've gotten almost everything I've worked on before was following everybody else saying it was impossible, and I was able to pull it off. So this is my track record, and that's why I believe that I can actually cure aging. And the reason I put that pitch at the beginning is the only obstacle to curing aging is the appropriate amount of funding. All the problems that I listed, I've been experiencing all myself. So what I've learned, a new way of thinking about everything from all this work that I've done of learning everything I can. I've learned that health, all aspects of health, is like a tug of war. We have mechanisms in our bodies for decreasing our health and increasing our health. And it's like a tug of war that's back and forth and back and forth. Now, most of us, or a lot of us, really just have this one-sided tug of war where we just have decreasing health. And as a result, we just get sick and sick and sicker and die early deaths. Okay, so there are things that we can do to decrease some of these things that are causing us to decrease health, but we really need to start focusing on putting people on the other side of this tug of war to increase health. And what we ultimately want to do is we want to win this tug of war with doing so many good things that we actually win the tug of war, which actually does not exist yet. Okay, something else that I've learned about the way to think about medical stuff is that we are just a gemisch. Now, I don't know exactly what this word means, gemisch, but it's something I've been using my entire life, and a Google search says the word doesn't exist, so I don't even know if I've spelled it right. But gemisch just means a big mixture of a whole bunch of things, and we don't know what's going to happen. This is a typical biochemical chart of all the different biochemical pathways in our body, and it's a gemisch, but it's worse than you think. Okay, and I want to zoom in on just one piece of this thing, and we can see that it's a biochemical pathway where one molecule gets acted by another molecule, becomes another molecule, gets acted by another molecule, gets acted by another. on and on and on. This is what biochemical pathways are. But if you are somebody like me that has done a lot of work looking at the biochemical pathways,
William,
looking at,
can I just
yes,
intervene?
yes,
Gemisch is a Yiddish word or German. It's German or Yiddish.
yeah.
It means a conglomeration of a bunch of different things.
How do you spell it?
Just how you did, Gemisch.
Okay, so I did
It's
spell it
a
right. I don't know why when I put that into Google, just to verify that I tried to spell it right earlier today, I couldn't. It said the word doesn't exist.
well, you have to say German or Yiddish. Bill
Okay.
knows this stuff. Consult Bill.
All right. I think I used the word at the
It,
Medical Excellence
it
Conference.
kind of means it's the small stuff. Don't bother me, and flick your shoulder. Don't give me a gamish.
So, I can't see any of you now with the
Okay.
PowerPoint up in front of me. So I don't know who's actually talking, but the but here's the thing is that it's a lot more complicated than this. If you're like me, I've done a lot of experiments to find out what binds to what, et cetera. Scientists do this all the time. This is how these biochemical pathways came up. They would look to see what binds to what. But when you start looking at what binds to what, you find out that everything binds to everything until you start increasing stringency. And then you find out what's the strongest binder. And then everybody forgets about all the other things that bind. So in reality, our biochemical pathways are more like this. We have things going every different direction, and it's really a gamish, and it's all this gamish that makes us human. So it's really hard to really understand biochemical pathways. You just can't look at it and figure out a cure. You have to dig deeper, do a lot of deep thinking about what's going on when you're trying to come up with cures for things. And let me just, some of this might be more complicated than needs to be for the talk, but two other things. One is gene regulation. The regulation of genes, turning genes on and off. That's what epigenetics is. And a lot of people treat this as too simple too. You have a promoter, which I like to call a dimmer switch because promoters can be turned on and off to different levels. And so what happens is some protein binds to that thing and turns the gene on. Okay, that's the very simple-minded approach towards thinking about this. But it's actually a lot more complicated. It's, again, a gamish of things, and all these things bind. And then just by looking at what binds and stuff like that, you have no idea what's going to happen. The only way to know is to actually do the tests and find out, and actually do diagnostics on humans to find out what is actually happening with that gene. Is it being turned on? Is it being turned off? And to what levels? Again, it's a gamish. Now, this is a really important thing here because this actually plays a big role, and this is something called enhancer sequences. And I'm sure a lot of you have never heard of these enhancer sequences because I think the only place you ever hear about it is in advanced molecular biology. But again, you have a gene, you have a promoter, the dimmer switch, but then you have something called an enhancer sequence that's a long ways away from this promoter. And the DNA will actually fold over, come in contact with this promoter with a bunch of other proteins and turn the gene on. This is a very, very important part of gene regulation that has made studying gene regulation very difficult. So a lot of people get real big misinterpretations of thinking that the genes are just too simple. They don't realize that sometimes genes are regulated by things far, far away along the chromosome from the actual promoter. But again, every DNA is an enhancer. Every single bit of DNA has the ability to bind proteins that can fold over and regulate that gene. And again, we don't know what's going to happen. It's just not that simple. And again, it's a gamish. But it's these kind of things that I've been thinking about my entire life that have actually made it so that I have succeeded at coming up with cures for some of these diseases when nobody else could do it. So, but my focus is on aging. I've been asking my entire life, what is aging? Why do we age? How do we age? How do we not age? And I want to make it clear, I'm not obsessed with this because I'm afraid of dying. I am obsessed with this because I think there's a lot of great things that are going to happen in the future. This is a scene from Star Trek First Contact. And when we actually make contact with life from other planets, I want to be there. It's not a fear of dying. It's a fear of missing out that really is driving me. And I want us all to be able to benefit from research to try to increase our ability to live longer and healthier. And if anything, what I want to do today is I want to show you that aging can be cured. There's no reason it can't be cured. It is something we don't have to have. Okay, now, after many years of working in other companies, I finally started my own company in 1998 called Sierra Sciences to focus exclusively on trying to cure the aging process. This is my website. You can go to that. It's also included in the attachment that I've included in the chats. But it's really important. 150,000 people die of aging on this planet every single day. And it turns out, there's studies, and the link to one of the key studies is shown below here. If we eliminated aging, half of us would still be alive and healthy in 1,000 years from now. This is how big a role aging plays in our diseases. And I want you to think about aging is not just a health-related thing. Aging is something that's a very programmed thing in our body. That's why aging is very synchronous. Okay, you can walk down the street, you know how old people are just by looking at them plus or minus a few years. And you can always tell, same with animals, you can look at a dog or a cat or something, you can tell that that cat's old or young. And the synchrony within humans is very consistent, but it's very different from the synchrony that occurs in other animals. So there's something programmable. The questions that I often ask myself is, why, if aging is something that's totally controlled by environment, why do we all age at the same rate, even if we live on the pole, North or South Pole, or live on the equator, where the environments are very different? And why do dogs and cats age at different rates than humans? Okay, so aging is something that I think is very controllable just because of the fact that it seems to be very controlled to something that exists in our body that's programmed. Okay, so the title of my talk is The What, Why, How, and How Not of Aging. And I'm not going to be able to talk about everything in less than 10 hours. So I want to direct you to my website, which is at www.sierrasci.com. And you can get a lot more details by watching key videos and documentaries where I go through almost every one of the things I'm going to talk about today in excruciatingly great detail. Okay, and my email address is bandrews@sierrasci.com. I actually love reading emails and answering questions. So if anybody wants to follow up on anything I talk about today, please don't hesitate to email me. And I'll provide those later again towards the end of this talk. So I first want to talk about the what. And the what is probably the toughest thing to talk about. I'm not going to go as much detail as I did at the Medical Excellence Conference, but there are the hallmarks of aging. Okay, and I don't like this idea of this is what aging is, because nobody has really explained what causes the hallmarks in most cases. So we have all these things that cause aging, but nobody has explained really what causes the causes of aging, except in a few of them, one of which I'm going to be talking about today. Okay, there's a lot of biomarkers of aging. A lot of people define aging just by the biomarkers. And yeah, we have all these biomarkers. There's 4 that have become really popular lately. That's telomere length, DNA methylation, IgG glycosylation, and total RNA sequencing. The bottom one's my favorite, actually, because it's the really best definition of epigenetics. It really is the best measure of epigenetics. But they're all pretty good at correlating with aging. Okay, the problem is that reversing a biomarker of aging. So if you're trying to use these to gauge your success at reversing aging, reversing a biomarker of aging doesn't mean that you have reversed aging. It only means that you have reversed the biomarker. And so when a patient comes into your office and says, I wish I was younger, I wish we could do something to make me younger, they're not saying that they want their telomere lengths longer. They're not saying they want their DNA methylation patterns changed or their IgG glycosylation patterns changed. They they want to, or their hand grip is stronger. They want to look, feel, and behave like they're 25 again. Now, we don't have a good measure of aging. The bottom line of what aging is we really can't, we don't know. Aging is not measurable. But we all know which photo of Betty White was taken first. Somehow we know that. Okay, we can remove the wrinkles, the fat in the skin, the hair color, everything like that. And we would still know that the photo on the right of Betty White was taken after the photo on the left of Betty White. This is her at 85 and at 25. And I'm sorry she's passed away now, because one of the big goals that I had was to actually be able to see if we could turn Betty White back into 25 again. And I'm serious when I say that. I really think it's possible, and you'll see why later on in this talk. Okay, but I consider that aging, even though we can't measure it. It's not quantitative, it's qualitatively quantitative. So here at Sierra Sciences, we use terms called QQ scores. You can take one picture, or you can have several different people, and you can put them in line in terms of age by qualitatively quantitating them. Even though you can't quantitate it. And we call that QQ, giving them QQ scores. Okay, this is what I call reversal of aging. When we actually turn somebody from the picture on the right to the picture on the left, and that's not just biomarkers. The biomarkers aren't going to do it. If you reverse the biomarkers, but the person and some patient, and that patient has not shown reversal like shown here in that picture, then aging has not been cured. Nobody in the history of the planet has ever passed this test. And aging will never be reversed until somebody actually does pass this test. I call this the Betty White test, and that is my gauge of success. This is how I'm going to gauge my success, and when I get the research completed to the point where I say I have reversed aging. There's a lot of products on the market now where companies are claiming to reverse aging, but none of them have actually passed this test. Okay, that's all I have on the what, because we don't really know. Nobody really knows what aging is. And this is one of the reasons why the FDA has not made it, call it a disease. Okay, I call it a disease. That's why I say I'm going to cure it. But it's like nobody has actually called it nobody can really define it as a disease until we can measure it. Next, I want to talk about why we age. And this is something I spent a lot of time talking at the Medical Excellence Conference. But why did we evolve aging? That's the big question. This is one of the reasons I did a population genetics background, PhD, when I was in graduate school. Okay, but more importantly, why did we never evolve a way not to age if aging is so bad for us? Well, here's the whole story simplified into just 3 different statements. But you can learn more, hear more about what I have to say on this by going to my website and watching the videos. But the bottom line is, the more diverse a species is, the more likely that species will survive rapidly changing environments. So evolution has all been about increased diversity within species. Now, it turns out that eliminating the longer lived adds diversity to the species. And that's because during sexual reproduction, we are shuffling genes. And when we have offspring, that offspring is a shuffle of our genes between 2 people, creating a gene combination that probably never existed anywhere on the planet before. Well, once you've done that, you now become longer lived. Once you've raised your young, you become longer lived. That's my definition of longer lived. I don't like to use the word old because life's about living. Life's not about getting old. Okay, so eliminating the longer lived as diversity, because after you've raised your young, there's going to be more diversity by letting the offspring interbreed than letting the parents rebreed. And as a result, a species is going to have more diversity if it eliminates the longer lived. So elimination of the longer lived, not just by aging, but all other ways of eliminating the longer lived, cancer, heart disease, all these different diseases that we've evolved or never evolved not to have, that result only or they're more common when we are longer lived than we're shorter lived. All these diseases, if we could, if by having them, helps our species eliminate the longer lived, which is beneficial to the species. Okay, that's all I want to say on the why. And you can hear more in my videos on the website, but now let's go to the how. Now, again, I'm not going to spend as much time as I did at the Medical Excellence Conference, but aging begins with wear and tear. But it's wear and tear that occurs during normal bodily functions to survive. Okay, we have to consume, we eat, we have to sleep, we have to do all these different things that require wear and tear of our body, our organs, tissues and organs. There's wear and tear that has to occur just to be able to survive. That's something we cannot stop. That wear and tear is going to be happening. And that's where that's where the synchrony comes from. It comes from the wear and tear that has to occur from normal bodily function. Now we can accelerate that with poor lifestyles and poor genetics, but bottom line is we have to have a minimum amount of wear and tear just to be able to survive. Now, this is the thing that kind of doesn't make sense at first, but humans have the ability to replace damaged cells. So even though the cells get damaged from wear and tear, we can replace them. And therefore, why do we age? Okay, and so we shouldn't age, really, if that was all there was to aging, just wear and tear. But the problem is that human cell division, at least cell division in humans, is limited. I got human in there twice. Is limited, and not only that, but every time a human cell divides, it undergoes changes that correlate with aging. Okay, so every time a human cell divides, even the epigenetics of that cell changes and is permanent. Okay, so something is going on there that's called, I'm going to explain here, is when a human is a fertilized egg, and I want to say this is not true in all animals. It's true in dogs, cats, horses, sheep, pig, and deer, humans, and primates. But it's not true for rodents and like rats and mice and rabbits. Rodents age entirely different than this. And that's one of the reasons why I think rats and mice are a terrible animal to be studying aging in. But when we are a fertilized egg, our cell can only divide 100 times. And a lot of people say, well, that's not enough times to create a human. But it turns out if you do the math on the 2, 4, 8, 16, 32 math, you would find that if a human cell divides 100 times, it'll make a mass larger than the planet Jupiter. Okay, so 100 cell divisions is quite a lot. But after the cell divides and becomes 2 cells, there's only 99 more times those cells can divide. Then 4 cells can only divide 98, 97, 96. When we're first born, we've already down to only 50 cell divisions left. And we still have a lot of cell divisions to go because we've got to grow up and stuff like that. 48, 43, 38, 20. When we get to be really old, we have less than 10. We get really longer lived, I meant. We have less than 10 cell divisions left to go. And this is actually a big, big problem with aging in humans and the other animals that I mentioned. This is called the Hayflick limit. It was discovered in the late 1950s, early 1960s. It was published in 1961 by Dr. Leonard Hayflick. Most people said this can't possibly be true. It took years before people started really believing it to be true. And now every lab in the world that works on human cells knows that it's true, because when you go to buy cells from a vendor, you have to specify how many populations does that cell have when you buy it, because you don't want to buy cells that are too old, because you won't be able to get those cells to divide enough times in your research. So you can actually ask the vendor how many times has these cells divided, and then you can buy the cells and then count them and verify that that was the correct number. There's something like ride tickets at an amusement park inside of our cells. This is something I realized back when I was still in college at my anti-aging clubs. And this is something that I struggled with for a long time. What could these ride tickets be? Because that was the only explanation as to how our cells could only have a limited number of cell divisions. How do our cells know how many times it can divide, or how many times it has divided, and how many more times it has left to divide, when cells don't actually have brains? So there had to be something like ride tickets in an amusement park inside of our cells. What are those tickets? And that was a dilemma that I bothered for a long time. But I want to say that no matter what else we do to cure aging, with all these other theories and everything like that about aging, no matter what else we do, our lifespans and health spans are still going to be limited unless we solve this Hayflick limit problem. This is the biggest problem there is in terms of aging, because we will never live very long if we don't, no matter what else we do, unless we solve this Hayflick limit problem. And it's the only cause of aging, theoretical or non-theoretical, that actually can say this. Okay, so the question is, why is human cell division limited? And we now know, long story short, it's due to telomere shortening. We were able to show this in the mid-1990s. It made headline news, et cetera. We were able to show that telomere shortening is 100% the explanation of the Hayflick limit. So what's a telomere? We just have to zoom in on a human body and then zoom in on the cells. I'm not going to go into much detail because I'm sure all of you know this stuff already. Chromosomes, the top arm and the bottom arm, each arm contains a DNA molecule that's tremendously long. The DNA is all folded up like a slinky in each of these arms. But I want you to think of these long DNA molecules as shoelaces. And we know that shoelaces, our shoelaces have caps at the end called aglets. And the aglets protect your shoelaces. Well, our chromosomes, the DNA in our chromosomes have the same thing. They're shown here in yellow. They're called telomeres. The telomeres protect our chromosomes. The telomeres are made up of DNA also. There's a very special sequence of TTAGGG. I'm sure most of you know that DNA is made up of 4 different bases, and the sequence of telomeres is a repeated sequence of TTAGGG. So if we unravel the slinky of the telomere part of the chromosome, we see that a telomere is about 15,000 bases in length, where the beads are called bases. And that's at least when we're first conceived. And this is where all the troubles begin because of the cell division that I talked about. Every time our cells divide, each and every time they divide, our telomeres get a little bit shorter. And so there's so much cell division going from the time we're a single cell embryo to a newborn baby that by the time we are born, we've already lost half of our functional telomeres, the part that I call the ride tickets. So we were conceived at 15,000 bases. When we're born, we only have 10,000 bases. But the problem doesn't end there because we still have a lot of cell division, a lot of growing to do, wounds to heal, infections to fight. So there's a lot of cell division. Our telomeres get shorter and shorter. And when our telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. And this is what is happening. This is not a theory. This is a fact. Every single lab that ever works on human cells in a petri dish knows that this is true. I'm going to go over that again. We are conceived at 15,000 bases. We're born at 5,000 bases, and we die of old age at, we're born at 10,000 bases, and we die of old age at 5,000 bases. And until recently, there's absolutely nothing we could do about this. It's an actual ticking clock. It exists in all of us. And it turns out this is where the ride tickets are. The ride tickets are found at the very tips of our chromosomes, and we lose a ride ticket every time our cells divide. We lose a little bit of telomere sequence every time a cell divides. So it's been now known from our work in our lab and several labs that telomere shortening absolutely puts a maximum limit on our lifespan and health span of 125 years. And this was reassuring because there had already been several publications, more recent publications shown below, showing that other measures using statistical theory and sociological studies and things like that, demographic studies, et cetera, have shown that other ways have shown that, yes, our human lifespan has a maximum of 125 years. And now telomeres explains why that is. It's the first thing that's ever come along that explains the synchrony and explains the limit on our lifespan and also explains why we see changes that occur as we age. Now, it's not just aging, okay? It turns out now that every disease you've ever heard of, except for just a handful of them, are now caused or at least associated with the shortening of telomeres. And in a lot of cases, the telomere shortening doesn't cause the disease, but it causes the symptoms of the disease. And I'm not going to go through today and explain all these things, but I do explain a lot of these things in a lot of my videos. And these diseases, telomere shortening diseases, are called telomeropathies. If you ever want to do a Google search to learn about any of these diseases, or PubMed search, let's say, but telomeropathies and the role telomeres play in diseases, just do a search using the word telomeropathies. Okay, the bottom line is bad things happen when telomeres get short. I know Sonny's on this call, Sonny Wynn, and he uses that term all the time. I used to use that a lot, but I'm getting back to using it now. All right, so that's pretty much the how. And so now the key thing is the how not. After everything that I've been studying my entire life, where has this led me on to figure out how not to age? Okay, well, first thing comes to mind is that we learned that telomeres don't shorten in our reproductive cells. And this was really important because if telomeres did shorten in our reproductive cells, our children would be born with shorter telomeres than we have, since reproduction requires cell division. And so since our children aren't born with shorter telomeres, they're born as telomeres as long as we had when we were born. That means that there's something going on in our reproductive cells that prevent telomere shortening. So we discovered in the mid-1990s that something in our reproductive cells is called human telomerase. It's an enzyme. It's only found in our reproductive cells. And this figure here is showing our DNA now as a green squiggly double helix. And this factory looking thing is human telomerase. It binds to the very tips of our chromosomes and it lengthens the telomeres. And it's so first the telomeres shorten and then telomerase re-lengthens them. That's what I want to show here. In our reproductive cells, I want to come back to this tug-of-war analogy. In our reproductive cells, we have shorteners and we have lengtheners. And we know that the lengtheners are telomerase. The only thing that's ever been discovered in normal human cells that can lengthen telomeres is telomerase. And you're going to hear a lot of vendors are going to tell you they have products that lengthen telomeres. Well, they don't really lengthen telomeres unless they produce telomerase, and I'm going to come back to that too. So in our reproductive cells, it's a tug-of-war that's a tie. And the key thing to show here is that first the telomeres shorten, then telomerase re-lengthens them. Telomeres shorten again every time it's cell divides, then telomerase re-lengthens them. So it's a two-step process. Now, in all the other cells of our body, there are no lengtheners at all. So we just have shortening occurring all the time through our entire life. So how do we add people to the other side? That's what I want to do. That's the approach that I am trying to do to develop a cure for aging. Well, there's two approaches we're taking. I'm not going to spend a lot of time on them. One is called gene induction. Since our reproductive cells contain the gene, since they produce telomerase, that means they contain the gene for telomerase. It's just the gene is turned off in all of our cells, as shown in a second here. When we go back, I guess it's not going to recycle here. Yeah, okay, so is turned off by this protein that's called a repressor, and we're looking for things that can bind to that repressor to dislodge that promoter, that repressor that's shutting the promoter off and turning the gene on. So that's what we're doing. We're looking for molecules of all kinds, mostly nutraceuticals because they don't require FDA approval, but that's what we're doing. We're looking for things that will turn the gene on. We're also doing something called gene therapy, which is where a gene therapy will fuse with a cell. The gene therapy contains the gene produces telomerase, and because we have the ability to engineer that gene, it produces a lot of telomerase. And so just think of these as two soap bubbles. The small thing is a gene therapy that we construct, and we can use that. And that's how we get most of our proof of principle concepts that we have shown that I'm not going to discuss today, but I will tell you they're in my videos. We have proof of concept showing that we can reverse aging in every way imaginable or measurable that you can ever think about in human cells in a petri dish, human skin grown on the back of mice, and also engineered mice. I mentioned mice don't age by telomere shortening, but we have created engineered mice that do, and we show that they age like humans, and then by inducing telomerase by gene therapy, we can reverse their aging. There's a video, Diane Sawyer interviewed a colleague of mine who is working on this, Dr. Rhonda Pinnell, and you can actually see all these mice, or you can talk to Sunny Wynn, who's on this call, and he has the video if anybody would like to see that. Okay, so, and you can learn more about these techniques. I go in a lot more details at my website, www.sierrasci.com. Okay, I want to come back to this slide because now it's a little more complete. No matter what else we do to cure aging, our lifespans and health spans will still be limited unless we solve the telomere shortening problem. Because it's not the Hayflick limit problem that's really the culprit. It's the telomere shortening that's the culprit. We have to solve that problem if we ever want to live healthy longer than 125 years. So now, what can you tell your patients? This is where everything's leading up to. Okay, well, first, you're going to have patients that want to have their telomere lengths measured. I'm the inventor or co-inventor of every method there is that exists to measure telomeres lengths. Also, telomerase I'm co-inventor or inventor of every method for measuring telomerase activity. But I can tell you that most people do not know how to do these tests. And the only test that actually I have any faith in, because of the difficulty in doing them, unless there's somebody in my lab who knows how to do these things, is a test that it's called fluorescence in situ hybridization, abbreviated FISH. And some people do this, but only one lab in the world will actually report what percent of telomeres that are short. And because of the fact that I'm not supposed to be talking about products and stuff during this call, I'm going to just tell you that if you are looking for a vendor, just look for a company that does telomere length measurements that actually measure telomeres by fluorescence in situ hybridization, and that they report the percent of telomeres that are short, because that is a much better measurement. The percent of telomeres that are short is a much better measurement than the average telomere length, because it's really most aging occurs when telomeres get critically short. So I'll leave it at that. Now, we can also do things so your patients can also be taught how to slow down accelerated telomere shortening. Okay, so, so accelerated telomere shortening just means that even though we have a maximum limit of 125 years, we can do things to accelerate that shortening, and that's inflammation, oxidative stress, things like that. So anything that reduces inflammation, oxidative stress, DNA damage, UV exposure, et cetera, all these things can be used to decrease the rate of accelerated telomere shortening. And I just want to go through some of these things. Exercise, I put that first because a lot of people think of exercise as something that increases inflammation and oxidative stress. But I'm big into exercise. I've gone to a lot of like ultra marathons and things like that. I've done blood work on runners before, during, and after the race to really start studying what's going on. And I've learned that exercise can be inflammatory if you're somebody that rarely exercises, and then when you do exercise, you exercise really hard because your body thinks that this damage to your joints, this pain to your joints and stuff like that is something attacking you. And so it induces an immune response, which is going to cause inflammation. But when you exercise every day and you keep it fun, you build immune tolerance, just like normal immune tolerance to something just as simple as exercise. And so your body does not cause an inflammatory response. And people can actually go out and run 100 miles and then run the very next day without any problems because they're not stiff as a board. If you're the occasional runner that runs every 2 weeks and then runs a marathon, you're going to do well, but you're going to be on your hands and knees throwing up afterwards, and you're going to be stiff as a board for 2 weeks just because of all the inflammation that's migrated to your joints. So just keep exercise fun, consistent, and fun. Okay, so also take your antioxidants, take your anti-inflammatories, tell your patients to do that at least. Control sugar levels. Now, there's a lot of ways to control sugar levels. There are products now that actually will convert sugar that you eat into fiber. And we know fiber is good for you. So these are kind of products that people can be taking right now to keep their sugar levels down. There's also reduced toxins. Okay, try to find foods that don't contain toxins, but there's also products that you can find that can actually remove toxins from your foods after you eat them. Just take these things when you eat, and they can reduce your toxins. Avoid inflammatory foods. There are actually really good tests that you can get now. These are not allergy tests. These are sensitivity tests for testing inflammation. You can actually get your blood tested for what foods cause inflammation, and just Google it. You can find these companies in Google searches, but don't focus on, look for inflammatory tests, not inflammation tests, not allergy tests, because of course IgE and IgA are very different. Maintain gut health. There are products that you can take that your patients can take that actually will do a really good job of maintaining gut health. Don't be obese. Obese and smoking, which I thought I had on there here. Both of those things are probably the best things to increase oxidative stress and inflammation in your body. Don't do those. Reduce stress, especially the kind that your boss gives you. So psychological stress. Reduce depression. Those things, all these things by doing all these things, you can reduce accelerated telomere shortening to the max. Now, so let me just discuss this again. Okay, so we have shorteners that are pulling our telomeres. They're pulling our telomeres to shorten them, but we can remove some of these people by antioxidants. So that's accelerated telomere shortening before we show them. Now we reduce the accelerated telomere shortening with the anti-inflammatories, antioxidants. We can do the same with anti-inflammatories. But it turns out there's a limit to how much we can do. There's something called basal level telomere shortening caused by something called the end replication problem. My videos explain this in great detail, but this is something that makes it so that we cannot get below what a basal level telomere shortening rate is, and that's what limits our lifespan to 125 years, our lifespan and our health span. So just bear with me because this goes slower because there's fewer people. All right, so now we can add people to the other side now. We have, especially from research done in my labs, we have found ways to do this. Now, we haven't won the tug of war, but we have discovered nutraceuticals, things like that actually will add people to the other side of the tug of war. And now people say, yeah, but you're still aging. You're not curing aging. Well, I want to make a really important thing. This is slowing down aging. This is a really good thing. And I'm just always shocked the number of people that say they're disappointed because we don't have something that actually wins the tug of war yet. The only thing that actually wins the tug of war is telomerase gene therapy, which I mentioned we use for our proof of concept studies, but it's extremely expensive and it's actually extremely risky. There are clinical studies underway, including some that I'm doing to look at gene therapy, but the only people that I would ever let participate in studies are people that have life-threatening diseases that are more likely going to have negative effects from not getting the treatment than from getting the treatment. Okay, but there are things that people can take now that are nutraceuticals that will add people to the other side of the tug of war and decrease the rate of shortening. Now, does this exist yet? As I just said, it only exists in gene therapies, but is there any nutraceuticals? Are there nutraceuticals that can win the tug of war? This is what we would really like to have, but not yet. Okay, they don't exist yet. And you're going to hear marketers telling you they have them already and things like that. And I'm going to explain in a few minutes how to know, how to be able to find out what's real and what's not real when you listen to these vendors. But given our research, we believe that we can actually win this tug of war. We can find a nutraceutical or something else that can win this tug of war within 1 to 3 years if we could get the sufficient funding. And this is one of the reasons I made the pitches at the beginning, is because the only obstacle that we have right now is sufficient funding. This research that we do to search for these nutraceuticals and stuff cost us $2 million a month on average. And it's very hard for us to complete that research. If we had that money, we could have this research completed in 1 to 3 years. And believe me, every penny would go into the research. Okay, so now I said companies say that they already have products that do this. Why do some companies claim that their products do lengthen telomeres? And here's why. Okay, so what they can do is they can show data that before they can measure the telomere lengths, even if they're using a good method to measure, they can measure the telomere lengths in your blood. They can show that they are short before you get treated, then after you're treated, they get longer. Okay, well, it turns out that doesn't mean that the telomeres got lengthened. There's other ways to actually make the telomeres in the blood longer without lengthening them. And that pretty much, because it comes down to just a redistribution of cells in your body. So like one example here, that I'm going to come with more in a few minutes, is when we have an immune reaction, cells in our bone marrow, naive immune cells and things like, what is the other word? I can't remember the other one, but naive immune cells in our bone marrow, which already have long telomeres. Even when you're 90 years old, you still have cells in your bone marrow that have really long telomeres. These cells divide and infiltrate your blood. Okay, and that's what makes them longer. One of the things that makes them longer. Okay, only telomerase lengthens telomeres. And so when somebody says they have a product, ask, did their product induce telomerase expression? And let me explain this a little more detail. So if somebody tells you that their treatment lengthens telomeres, the first thing you want to do is you want to ask if their treatment expresses telomerase, because it doesn't lengthen telomeres if it doesn't express telomerase. If not, it does not lengthen telomeres. Okay, now we here at Sierra Sciences, because of the fact there's 0 telomerase produced in all the other cells of our body, we can't, there's a bunch of changes occurring in my slide here. We cannot say how much, we cannot gauge our success in producing telomerase in our cells by saying how much fold over background do we have, since the background's 0. Okay, so any kind of expression, any kind of production of telomerase, if a background is 0, is going to be infinite expression. So because you've got the division by 0 problems. So instead of measuring by fold over background, we look at the amount of telomerase produced in HeLa cells. HeLa cells is a very famous cancer cell line. And we look at this because it just so happens that HeLa cells express just as much telomerase as needed to tie the tug of war. So it produces just as much telomerase as our reproductive cells do that tie the tug of war. So there's a few red lines that just popped up on my slides. I don't know where those came from. Oh, now they're going to stay with me. Okay, so now, so if somebody tells you that their treatment lengthens telomeres, ask if their treatment expresses more telomeres than HeLa does. If not, it does not lengthen telomeres. Even if it did produce any telomeres, it's not going to reverse aging or lengthen telomeres and by reversing. It's not going to lengthen telomeres to reverse aging unless it produces more telomeres than HeLa does. But you're going to find out that nobody has any data showing that they are producing any telomeres at all. Okay, so I'm going to explain it in a little different way here. Okay, so we have a lot of different types of cells in our blood, but every single type of cell in our blood is also a mixture of cells with different telomere lengths. Because the telomere length in any blood cell, except red blood cells that don't have any telomeres because they don't have nuclei, all the white blood cells, the length of the telomeres in each cell depends on how many times that cell has divided before it got to where it is. And some cells have divided more times than others. So you have a big distribution of telomere lengths in your blood. This is one of the things that makes it really hard to measure because most companies, all they do is report the average telomere length. And as a result, you think all your telomeres are the same length. Okay, but since we have this big distribution, don't think that when somebody reports your telomere length that all the cells in your blood have the same telomere length. Okay, so if we remove cells from your blood that have short telomeres, the average length of your telomeres increases. And that's just like if, let's use an analogy of a company. If you have a company and you fire your dumbest employees, the average IQ of your company increases without anybody getting any smarter. Okay, so the same thing is happening in our blood. If we remove cells with short telomeres, the average length of the remaining cells in our blood increases without any lengthening occurring. This is a publication from PubMed showing that any product that is toxic at all will preferentially kill the cells with the shortest telomeres because the cells with the shortest telomeres are more sensitive to toxins than cells with longer telomeres. So every time you do anything that's kind of toxic at all to your body, it's going to increase the average length of your telomeres. But because other cells have to divide to replace the killed cells, the average telomere length of your whole body has decreased, even though the blood telomere lengths have increased. So this is one of the explanations as to why a lot of companies claim that they're lengthening telomeres. They're not. Their product is just removing. One of the explanations, their product is removing cells with short telomeres without lengthening and causing the average length of the remaining cells in the blood to be longer. Same thing happens if you were to add new cells to your body, to your blood that have long telomeres. As I mentioned before, when you have an immune response, cells in your bone marrow divide, infiltrate your blood, adding cells to have long telomeres. Again, your average telomeres are going to increase in length without any lengthening. It'd be the same if your company hired geniuses. The average IQ of your company increases without anybody getting any smarter. The average length of our telomeres will increase without any lengthening occurring. Here's a publication that's showing any product that induces an immune response is actually going to add new cells to your blood. Okay, so bottom line, a treatment that is toxic or immunogenic is going to do the same thing without any telomere lengthening. You need to measure telomerase expression if you want to believe that your product is actually lengthening telomeres. And this is what you have to ask the companies, the vendors that are selling products, they're claiming to lengthen telomeres. You have to ask them, do they have any data that's showing that they are inducing telomerase expression? And if you find any, please let me know, because I would like to see their data. And I would almost certainly tell you that their data is not what they think it is. Okay, so We do test every product here at Sierra Sciences. And when we get a result, we do let the companies know. And so look for our Sierra Sciences or the Bill Andrews seal of approval when some company says that they measure it. Because if some company says they have a product that actually does lengthen telomeres, we will let them say that, let them show our data showing it. Okay, now I'm going to finish up now. Things I didn't spend time talking on. There's a lot of rumors that used to exist. They hardly exist anymore, especially since 2017. There's people that believe that telomerase causes cancer, but it does not cause cancer. It actually decreases the risk of cancer by boosting the telomere length in our immune cells. Our immune cells are our best defense against cancer, but it also keeps telomeres from getting short in our cancer cells. That actually, in the short telomeres in our cancer cells cause skyrocketing mutation rates, which actually causes the cancers to mutate to a form that produces its own telomerase. So if we can keep telomeres long in all of our cells, it'll decrease the risk of getting cancer. And if we keep telomeres long in our cancer cells, it'll decrease the chances of them mutating to become immortal by inducing telomerase, and it will keep them from mutating to forms that will become resistant to whatever treatment you provide. You can learn more about this by going to pages 64 to 77 of my book called Telomere Lengthening. It was available at Amazon.com, but it's just too much trouble to keep it going there. So if anybody wants to get a copy of the book, just email me and I'll send you a PDF of it for free. But there's also one of the videos that you'll find on my website is a video where I spoke at a continuing medical education conference in Tokyo in 2017. There were 300 doctors in that audience, a very high profile presentation. My picture was even on billboards. When I got off the plane, I was surprised to see my picture on billboards. But the night before that presentation, somebody decided to get all over the news saying that telomerase causes cancer. So I stayed up all night, added some slides to my presentation that began at 25 minutes and 25 seconds into that presentation, where I, beyond a shadow of a doubt, proved that telomerase does not cause cancer. And you can watch that video, just begin at 25 and continue watching until 1 hour and 2 minutes, because that's how long it took me to show all the data that proves that telomerase does not cause cancer and actually decreases the risk of cancer. Okay, so there's a lot of things I didn't discuss. For more on telomere biology, again, go to my website. There's a website and click on that button. Proof of concept experiments, I mentioned them, but you can see the data. You can actually see the experiments that were done to proof of concept using the gene therapy approach. You can see aging mechanisms that exist in other life forms. I mentioned that there's only a few animals that age by telomere shortening. What causes aging in all the other animals? Well, they do have aging processes, because it turns out it's still beneficial to eliminate the longer lived for every species. It's just that not every animal eliminated the longer lived by shutting off the telomerase gene. So you can listen to my explanation of all the other animals. You can look at the mechanism of action for why a telomere length puts a limit on our lifestyle. Why does the short telomeres actually put a limit on our lifespan? I have videos that explain that in excruciating biochemical and molecular genetic detail. There's also a mechanism of action for why we see changes in everyone as we age. Okay, so when the telomere gets shorter and shorter and shorter, why does that cause changes? And I'm going to tell you right now that it's because telomeres are enhancer sequences. I mentioned enhancer sequences before that fold over, come in contact with genes, and turn the dimmer switches, the promoters, on and off of our genes. Well, telomeres are enhancer sequences. The difference between telomeres and regular enhancer sequences is that telomeres get shorter and so can't reach as far. What causes basal level telomere shortening? Okay, I didn't go into that detail, but why? Why did the telomere shorten every time a cell divides? You can learn about what all that is by going to my videos. And then there's a lot of data from these products that we developed that only add one person to the lengthening side of the tug of war. But there's a lot of data coming out, anecdotal data but I don't always believe anecdotal data, but some of this data is really difficult to believe, especially the results we're seeing in some of the older patients. Okay. We are seeing some age reversal when the telomerase is only producing enough when the genes are only producing enough telomerase to put one person on the lengthening side of the tug of war. Well, now we know why that is happening. And that is explained, and it has to do with the fact that when telomeres get critically short, there are fewer people pulling to shorten the telomeres. Okay, there's the analogies I use, they're falling off a cliff. I also use an analogy explaining the telomeres like a ball of yarn, and the telomere is the tip of the telomere is in the middle of that ball of yarn. And when the ball of yarn is big, it's hard for telomerase to get to that tip to lengthen it. But when the telomere gets short, that ball of yarn gets smaller, and it's easier for the telomerase to get in and lengthen it. But probably went over that too fast, but watch the videos. And there's much, much more in there. So my vision, living's the greatest thing that ever happened to us. We all are taking it for granted, and we only get it once. Living is just the greatest thing that ever happened to us. I love living. I don't want to ever see living become any less lovable for any of us, including myself, just because of this dumb thing called aging, which I think does not have to occur. We know enough about aging that we know it just exists for the survival of our species. Well, now we're smarter than evolution. We can undo this thing that we evolved or never discovered a way to not evolve because it was beneficial to the species. So that's my vision. My motto is cure aging or die trying. Thank you. And I'm just going to leave that up while we see if there's any questions. So if anybody hasn't had a chance to write down my website or email address. All right. So I guess I can't leave this up because I got to stop the share now. Let's see.
Yeah, lots of questions here.
Okay. Right.
We got Helen from Helen Care, Vietnam. May I ask how many times of multiple may be accepted? I'm not sure what that question is. So,
are these are not on the chat?
Yeah, they are.
Okay, I don't see them on my chat. Let me just. Oh, now it just popped up. Okay, now I see 23.
I'm not sure, Helen, if you're still on with us.
That question was about how many divisions of the cycle that you talked about.
So our cells can only divide a maximum of 100 times. Okay. And by the time we're born, there's only 50 cell divisions left. And that's not a theory, that's a fact. And I actually, in some of my videos, I actually show pages from vendors where you actually specify the age of the donor of the cells you're working with and the number of times those cells have divided since then.
Another one, I'm not quite sure what the question. Do we know if our children have long telomeres? They are expected to live a shorter life than us?
If they have long telomeres, they would live a normal life.
Correct.
Everybody's born or conceived at 15,000 bases and born at 10,000 bases. There are exceptions, especially the disease called Huntington-Gilford disease, where children are born, conceived and born with short telomeres. And they end up dying of all the age-related ailments that normal old people do when they're 20 years old. Okay, there's the pictures of these people are horrible. The research that I'm doing would be a cure for these diseases. A lot of people studying the diseases look for problems within these children, but it turns out the problem's actually in their parents. Their parents had a defective telomerase gene. As a result, the sperm or the egg had short telomeres, and these children ended up being conceived with short telomeres, even if they did not inherit the problems with their parents' telomerase gene.
Okay. Dr. Antoli has her hand up. Are you there?
Yes, I wanted to say that's such amazing presentation, and it's not even, I don't think it's that much money for the, I mean, this is a breakthrough in in science. I mean, to be able to make people live longer and all this reverse aging. That's unbelievable.
You would think so. You would think so, but I've had 50 plus investors. I'm sure glad to see Michael Gerber here. I just see his picture for the first time. How you doing? Um, I've had 50 plus investors. All of them were more interested in a quick return on their investment than they were in actually getting a cure for diseases. All of the investors, they're all uh, they're focused on trying to make quick return on their investments. They all think they can solve the aging process later on. And as a result, even though we succeeded at getting them their quick return on investment, they wanted to sell the company. And this would have sold, lost all my IP and everything. I was lucky to find ways to get out from under all that. And now I own 100% of my company. And I'm only going to be looking for people that really need me to cure their aging and believe I can cure their aging.
Well, that would be all of us, wouldn't it?
Well, yeah, all of us may be here, but
You don't get out of the. So let me ask you this: I took one of those True Age tests. They're supposed to, tell you how your insides are. They told me my telomere. I'm 72. My telomeres, I'm 55.9. Actually, these tests, they could tell you anything. How do I know what's in them? But ironically, I don't even know. My interest is in hormones. My hormones, I'm 79. That was my worst category. Are those tests of any use?
Yeah, I do think that True Diagnostics, what markets True Age, it's run by Ryan Smith. He's the founder of the company.
Mm-hmm.
I do believe he's one of the smartest people I've ever met in terms of this, but they're only measuring aging, using tests that correlate with aging, and they're actually not measuring telomere lengths because telomere lengths are very difficult to measure correctly. They're actually, and this is something that I've promoted a lot, is it's actually the DNA methylation patterns actually correlate very well with telomere lengths. So you can actually predict the telomere lengths from the DNA methylation patterns. So True Diagnostic measures your methylation and your DNA, not your telomere lengths. But since you mentioned a product, I'm going to take the opportunity to mention.
Yeah, no, we're good. Go ahead.
The company that actually is the best for measuring telomeres is a company called LifeLength. They're the only company in the world that actually uses, well, other companies use fluorescence in situ hybridization, but they're the only company that actually looks at each cell with a microscope, looks at each telomere with a microscope, and then reports the percent of telomeres that are short. They're located in Spain. That's the only place, but you can still get tested by them because they have companies all over through the United States that will collect your blood. They'll even send a phlebotomist to your office or home, collect your blood, send it to Spain, and give you your measurement. But that is the best commercial way of actually measuring your telomere lengths.
What was the name of that again?
LifeLength, LIFELENGTH, 2 words. And then their website is www.lifelength.com, and LifeLength's just 1 word there in the website.
Okay, great. Okay.
I think the previous woman still had some more questions. I interrupted her.
Okay. Dr. Antoli, do you have any?
I just want to say that I attended a conference with TA65 in Manhattan, and the doctor who presented, he's the founder of this company.
Joe Raphael, the founder?
Yes. He said that if we get the infection, a viral infection, our telomere shortened by half. And he told us also the senescent cells decrease if we have longer telomeres. So because our immune system is affected because high percentage of the cells are senescent cells. And he works with a younger man who comes at the conferences, and he told me that he takes this product, and he looks younger.
Yeah, you're probably talking about Joe Raphael.
Yeah.
Dr. Joe Raphael.
Yeah. Mm-hmm.
Well, he's not the founder. He's a doctor who works with the company. I used to work with TA Sciences, who sells market for TA65. I'm one of the key inventors of TA65.
Wow.
But
Thank you, Kevin.
I want you to ask them to show
Yeah.
you their data that shows that they produce telomerase. Telomerase.
I see. Because he had he works with some labs in California, in LA, that they check the length of the telomeres. But I did not. He gave us some PDF. He gave us some USB that we can put and look at all this. But I didn't have time to do it. I'm sorry.
Did you get the USB at A4M?
Yeah, I met with him first time when I was in Charlotte in for IV triangulation.
I've got that same memory stick right here. But the if you open it up, you'll see that my name is listed on the very first publication for the discovery of TA65. But it also showed data figure, I think it's figure 3 in that paper, is my data showing that I was able to detect telomerase expression from TA65, but it was, but you have to look at how much is being produced, okay? And now they have since published a lot of data saying that they're lengthening telomeres, but all they're doing is they're seeing that their telomeres are longer after treatment than before, and only in the blood. Okay, and so that's why I just mentioned that thing about you can have telomeres longer after treatment than before by anything that's toxic or immunogenic, because it actually causes a redistribution of the cells, but without any lengthening. So I'm not going to say anything more because I don't want
Yeah, I understand.
lawyers coming to me with cease and desist letters. But
Yeah, I want to say something very important. If you that there is we in our blood. So you said that after birth, that cell divides only 50 times, but which cell specifically? Because some of cells they divide every 7 days, some of them every 100 days. I mean, they have different.
that's not true though. There's a lot of rumors going around, but people aren't taking into account the 2, 4, 8, 16 math. Okay, people keep telling me, yeah, we lose 100 million cells from our skin a day. How's that possible if our cells only divide? You know, our cells divide typically once every 2.5 years. Okay, and they don't divide every 7 days. That's and anybody working with cells knows that the only way to get cells to divide that often is to really use a lot of mitogens and stuff like that, which would be very lethal to a human if they took those mitogens themselves.
Oh, really? That's amazing. And the other thing is that there is a product. The embryonic stem cells in our blood, we have embryonic stem cells, and those reverses ages by 2.5 years when we activate them.
We have stem cells in our blood. We don't have embryonic stem cells in our blood.
It's called the embryonic-like stem cell, VSEL.
Okay, you're using a terminology I'm assuming some company has invented to make the product sound better. But the only cells in the entire human body that actually do produce telomerase are human embryonic stem cells. And when I was working at Geron Corporation, Geron was one of the key companies that was involved in the discovery of human embryonic stem cells. At least when I was there, we funded the labs at the 2 universities, I think in Wisconsin and somewhere else, Johns Hopkins, that actually did the discovery. Yeah, there's a lot of misinformation being circulated. And I would recommend, I do a podcast called Up One, where I do what's called critical meta-analysis of peer-reviewed studies, not just meta-analysis, but I go in and I analyze papers and research, look at data analysis, experimental design, construct validity, statistical theory, look at all these things and really determine whether papers are real or not real. Because remember, the publish or perish. People are trying anything they can to get the number of publications up because their gauge of success is the number of publications that they have. So there's a lot of misinformation out there in the scientific peer-reviewed literature. But I would recommend send me an email.
Yes.
And I can follow up on a lot of this stuff because that's what I'm really good at. And that's what my podcast is all about. Go look for the Up One podcast with Bill Andrews. So Bill Clearfield, can I ask a question?
Yes.
You just mentioned TA65.
Yeah.
Can
Yeah,
I?
that's fine. Go ahead.
Okay. So the best product that I have ever discovered is called TeloVital, and it is available through a company called Touchstone Essentials. But it has the most potent telomerase inducers that we've ever discovered in that product.
Problem is you're always out of it.
Well, yeah, that's the problem is that
That's the problem. I
we
tried
did
ordering it myself and I couldn't get it. You
we did so well at A4M.
tell
Thank-
Sonny
win-
tell Sonny we need some more.
Yeah, well, Sonny, are you still on the call? Can you provide any clue as to when it'll be available again? Because I don't market it. I just discovered the.
Tell Sonny to go in the go back there and make a new batch.
Are you allowed to tell people when it'll be available?
Yes. Yeah, we're good here.
It will be available next week, and Dr. Clearfield, I will be letting you know. You'll be the first person to know about it when it
All right.
launches. Well, thank you so much for all you do. Appreciate you.
Thank you. What effect does high dose IV vitamin C have on telomeres? Is the question.
Well, vitamin C is really, really good. It's a great antioxidant, but it has no effect on telomerase, but it does decrease the rate of accelerated telomere shortening due to oxidative stress. Oxidative stress is a great way to accelerate the shortening of telomeres. And my videos explain how that happens too.
Okay. What products transform sugar into fiber?
Okay, so Gluco Control, also sold by Touchstone. And let me just qualify. So I should disclose, I do get a royalty from Touchstone Essentials. My company gets a royalty from the sales of Touchstone Essential, from TeloVital only. And we use every penny of that to fund our research. So it's not like I'm putting a lot of money in my pocket as a result. But I don't get any commission or any compensation from any other product. And so when I say that they have a product called Gluco Control that is really good because it converts sugar into fiber, I believe it. I've done due diligence on all their products, and I am one of Touchstone Essentials' biggest fans because I've never seen a company with a better array of products that really are really healthy for you. And again, I don't get any compensation for saying that whatsoever.
So where can we fit that in a diabetic diet regimen?
Well,
would that be appropriate or not?
Well, it depends on. Well, actually, it would be in all cases. Yeah. So, just maintaining proper sugar levels is the most important things in diabetes. But yeah, diabetics are not going to want their sugar levels to get too high. So glucose control is a good way to keep the sugar levels down. But also, just consuming less sugar will do the same thing. I'm trying to think of other ways to contribute there. But yeah.
I mean, what about like berberine? We use Gymnea sylvestri. There's another one that we use. Something with a B also.
I know berberine's good. I'm not familiar with the second one you just mentioned.
Use Gymnea sylvestri seems to curb sweet tooth for some reason. So.
Cinnamon, alpha lipoic acid, ginseng. So it's all like blended in supplements.
Well, all the things you just mentioned are really good things, whether you're a diabetic or not a diabetic.
Mm-hmm. Okay. Someone's asking about Protandim. It's stating reduces oxidative stress by 50% in 1 month. With a question.
Yeah, it works through the NRF gene. I think Protandim's a good product. Though they do make claims that it lengthens telomeres, and you just need to ask them, does it induce telomerase expression?
Okay. All right, dude. Here's somebody who's asking, they have Hashimoto's from time to time. They have MAP tongue. Any experience with this?
What's MAP tongue? I'm not for. Any-
In Chinese medicine, the tongue has all sorts of patterns that you're
Oh,
looking
oh,
at.
Matt,
You're looking at various organs in various locations,
Yeah.
the energies in the locations on the tongue. I'm not sure if maybe that's what she's
Yeah,
speaking.
and I don't know the answers to those questions.
Okay. And then there's a product specific astragalus from Mongolia that supposedly lengthens telomeres.
Yeah, ask them to show the data that's showing it lengthens telomeres. I'm the person that discovered that astragalus actually can induce a little bit of telomeres, but nobody's ever been able to even reproduce that data because it was so difficult to measure. It was so
My
low.
son lived in Mongolia for 3 years. He'd tell you to look at any of that kind of research with a little bit of a jaundice eye. That much I know. There are labs that sell cells without multiples. There are 3 to 12 times. Again, I'm not sure.
They're probably cancer cells. I assume they mean when they say without multiples, they mean with that can divide indefinitely. That means they're cancer cells or
Okay, that's our participant from Vietnam again. So if you're still on, Helen, let us know what this one's about.
I'm still here.
Yeah, okay. You wrote here there are labs, they sell cells without multiples. There are 3 to 12 times. So I don't know if you can elaborate on that.
Because I also get cell from US and their lab they say they sell like origin, not they don't multiple. And there's some labs in Asia, they multiple 12 times. So I don't know which is better.
what does multiple mean?
Did you find it?
When they replicate.
Oh, okay. So they sell cells that don't replicate, is what you're saying?
Yes.
Okay, so they don't replicate. There are labs, they sell cells that don't replicate.
Yeah.
There are 3 to 12 times. What is the three?
I mean, there's labs in the US, they don't replicate, and there are labs in Asia, they replicate the cell 12 times.
And so they replicated it 12 times, and then what?
And then I don't know which is better. Whether I use the origin, and then they say it's when it comes to the body, and then it gonna multiples replicate inside. And some labs they replicate it, and then so that the number of cell is more.
Oh yeah, well,
For-
when you replicate, you're going to have an increased number of cells.
Yeah.
I don't know if I'm going to be able to understand your question. What did you mean by origin?
It means they never replicated.
Okay, so you get, well, all cells have to have replicated to become a cell. But then they divide, so you're saying the company divided them 3 to 12 times and then sold them to people? I'm sorry. Maybe we should, you want to email me and maybe I can help figure out the answer to that question? If I understand the question better.
Okay.
Okay. Since women have had more reproductive problems and sperm count has plummeted, is it possible that telomere length in our
Nãy giờ xài được bao nhiêu?
reproductive cells are no longer long?
Cái này.
I realize there are many reasons for this,
Đúng
but
không?
could
Bỏ
that
vô đi. Lấy cái
explanation,
này thôi.
Somebody's got to mute because I can't. Okay, say it again. Are you reading
I'm reading this here from
Jeanette
Janet.
Cerna's
Yeah,
question?
Jeanette. So
By reproductive problems, you're meaning giving birth, not cell divisions. And
that's what that
sperm
sounds.
count has plummeted. Most women have more reproductive problems and sperm count has plummeted. Like is in our reproductive cells are no longer long. Okay, so that can be the case, but it's not. Okay, so in the production of our egg cells up until meiosis. Okay, there's telomerase is turned on, so there's no telomere shortening. Then when cells go into meiosis, there's no more DNA replication, so there's no telomere shortening. Okay, so the eggs and the sperm are all the same telomere length of 15,000 bases. And then when they're used to fertilize the egg, when the sperm fertilizes the egg to produce the embryo, that embryo has the telomere length of both those parents' sperm and eggs. So they're 15,000 bases. I did a 3-hour presentation at an infertility clinic one time on this subject. There are a lot of things that can reduce the rate of can increase the telomere shortening and things like that in our reproductive cells, but it's rare. And but even in everybody, there's going to be some sperm and some eggs that have short telomeres. And so one of the things I talked about was ways to be able to identify which egg and which sperm have long telomeres and use those for the fertilization. But yeah, there is, that's a pretty, let me read the rest of the question. I realize there are many reasons for this, but could you explain why?
Okay, she got it. Went to spam.
Okay, so I think the reason why our children are predicted to live shorter lifespans than us has nothing to do with telomeres. It has to do with environment and health and everything like that. And lifestyle, mostly lifestyle, obesity. What's another question?
What products from Touchstone are good for Alzheimer's disease or senile dementia?
Oh, well, Alzheimer's is a real difficult one. That's something that I study a lot. That's one of my clinical studies that I have listed in clinicaltrials.gov that's still trying to get underway is dealing with. But when we did the mouse experiments with the engineered mice, we found that mice that had become demented and were unable to remember how to go through a maze did remember how to go through a maze again after we lengthened their telomeres. So it's kind of suggesting that Alzheimer's isn't the loss of memory, it's just loss of access to the memory, and lengthening telomeres could restore that access. So that's one of the reasons why one of my clinical studies, clinicaltrials.gov, is looking specifically at Alzheimer's, because I think it could be a cure for Alzheimer's. But there's so little, so many rumors going around about what Alzheimer's is. All these new antibodies that have made that attack the beta amyloid plaques or microfibril tangles and things like that, but none of them actually decrease the symptoms of Alzheimer's. I don't really know anything that anybody can do except on the psychological level. I do believe I've seen a lot of studies showing that keeping your mind active, doing especially math problems, the number one best test is to repeat a sequence of numbers backward. When somebody tells you a bunch of numbers or you read a bunch of numbers, look away and then repeat them backward. That involves both problem solving and memorization, and that keeps your mind working. One of the things, so I'm a scientific consultant for a company called Synap that's also struggling trying to get funding, but I think they've got a really, really great way of possibly relieving Alzheimer's, and that's there's a protein called NOGO-A that exists in our cells that actually inhibits, it's NOGOA, that inhibits more neurites from forming. When your neurons need to reach out and connect with more neurons, they form neurites, and NOGO-A inhibits neurite formation. I mean, it's silly. Why would we have such a thing in our brain to do that? And that comes back to our reason why we evolved an aging process. It's good for the species to eliminate the longer lived. Well, so they're working with me to try to come up with inhibitors to the NOGO-A so that we can restore neurite connections and mostly brain activity in Alzheimer's patients. But their first clinical studies are going to be on stroke patients. because that's one of the, they believe that people with brain damage resulting in stroke could actually, people could recover by being able to induce neurites to grow. Yeah, I mean, things like, what is it called? There's been a bunch of product, oil from coconut, so coconut oil. There was a lot of studies on, a lot of press about coconut oils reversing Alzheimer's in patients, but that actually turned out to be a big scam. A woman published a book, made a lot of money, but Trader Joe's used to make mountains of piles of coconut oil in their stores, and now they don't have them anymore. There's been so many different things that have come out that actually been touted to reverse Alzheimer's, but nothing works. We know very little about Alzheimer's still, except I believe the best thing we can do to defeat Alzheimer's is lengthen telomeres. And that's mostly in the oligodendrocytes and the Schwann cell, well, not the Schwann cells, because that's peripheral nervous system, but the oligodendrocytes and the astrocyte cells. We could lengthen the telomeres in those cells and the neurons too, which they don't divide, so they don't really have telomere shortening except by accelerated telomere shortening. But I believe lengthening telomeres is probably one of our best approaches towards dealing with the Alzheimer's problem.
So Dr. Siegel, our integrative veterinarian, wants to know if you have anything for telomeres testing for dogs, cats, horses, and any products for them.
Yes, this is a very important subject for me. It's been pet products is high on my list. More people spend more money on their pets than they do on themselves. But I have been, if you watch my videos, especially one that I did in 2019, you'll hear me at the end talk about a product called Telo Dash, T-E-L-O, and a dash, and then the words dash, D-A-S-H. That's named after my dog, whose name is Dash. But it's essentially a salt shaker that contains telomerase inducers that you can sprinkle on dog food, cat food, or horse food. I mean, it's not going to work on your rabbit food for your rabbits because they don't have telomere shortening and they have telomerase produced in all their cells already. But I'm working with a woman. Her company doesn't have a name yet, but she's actually taking this on, and she's actually just gotten some funding and gotten some product made, and she should be launching Telo Dash pretty soon. I was hoping by now, but when Telo Dash gets launched, believe me, it'll be on my website. But I think it'll be the best product ever for pets, just like I think Televital is the best product ever for slowing down the aging process in humans. It's going to, the same kind of thing is going to happen with Telo Dash for pets.
Okay. There's a bunch of questions and suggestions about autoimmune thyroid disease. Get in touch with us. We have some videos on our website, aosrd.org, and I think we have a pretty good handle on it. We have I probably have about at least 150 active patients with it, and I've got it down. The endocrinologists in town absolutely hate me. So we're over the target. So and we have a video. Go to like I said, aosrd.org, and just do an open Apple find or an open thing find and type in Hashimoto's or my name, and you'll find the video there. Okay.
Michael Gerber has his hand up.
And we can do that too. So Dr. Gerber has his hand up.
Yes.
Dr. Gerber.
Well, yes. Thanks for your presentation, Bill. Very, very great as always. I was just wondering if you've ever looked at telomerase enhancement with hormone replacement therapy, thyroid, adrenal, sex hormones. Have you taken a look at those guys?
Well, I haven't, but other people have. And that's something that we discussed in this conference I did in that I mentioned before. I talked for 3 hours in Bogota, Colombia, at a clinic called Cecofes, C-E-C-O-L-F-E-S. But there's mixed data, but I do believe that any kind of hormones and stuff like that are going to be controlled by the level of the length of telomeres. And we have shown, which one did we show? We have done experiments. Well, no, that wasn't a hormone. That was, so we've looked at several different genes in vitro, in a human cells in a petri dish, where we can lengthen, shorten, lengthen, shorten, lengthen, shorten. And we've seen different proteins go up and down, up and down, up and down, directly in proportion to those things. And SIRCH1 is what I was thinking, but that's not really a hormone. SIRCH1 expressions go up and down and up and down and up and down with telomere length. I'm trying to think of what else. Yeah, but I don't know anything in particular, but I can't wait to be able to do the studies. We just need to really do the studies. We have to do them in humans, and we don't want to do gene therapy to do them in humans. We want to do it with a supplement that actually will be potent enough to win the tug of war in humans.
Thanks.
Okay, thank you. Dr. Nario, do you have any questions online?
Yeah, so I think Dr. Bill answered this question real good. A lot of questions about cancer, because there's a misconception that cancer cells hijack telomerase to becoming immortal, right? This was the initial thought, but I think you did give us some good resources to follow through in your website, I think.
Yeah.
I think this is oh, go ahead, sorry.
Let me just comment that so cancers do hijack telomerase. By hijacking means that when a cell becomes cancer, every cancer is caused by mutation. Okay, and I've had numerous debates with a lot of people about this, and nobody's ever proven it. Okay, so cancers start with mutations. That what a cancer is out of control cell growth. Okay, now remember, I was National Inventor of the Year for my cancer research, so I actually know the subject really well. But when you induce telomerase, it doesn't cause the cells to divide. All it does is give them more ride tickets. So it gives them more opportunities to divide when they are supposed to divide. Okay, so, but a lot of people started thinking that, boy, you're inducing these cells to be cancer by inducing telomerase. But no, it's not. The cells are still in control. Now, so like you'll never find a normal human cell that produces telomerase, except for our human embryonic stem cells. But you'll find that sometimes a cell becomes cancer, it loses growth control and starts dividing uncontrollably, or with less control than it did before. And as a result, the telomeres get really short faster. And when telomeres get really short, that induces a lot of skyrocketing mutation rates through processes called chromotherapis and catagenesis, it's called, where it's like when, like a lot of my cancer research, looking at human cells in petri dishes, in microscopes, I can actually see the mutations because most of these are chromosome rearrangements. The mutations are so big. This is induced by critically short telomeres. Okay, so a cell becomes cancer by a mutation, starts dividing, telomeres get really critically short. It's approaching its Hayflick limit, but because of the skyrocketing mutation rates, some of these cancers mutate again to induce telomerase expression. And they would have never mutated to produce telomerase unless the telomeres got really short. So keeping the telomeres long decreases the rate of the mutations that have cells ever become cancer, and also decreases the rate of mutations ever inducing telomerase expression. And so I'm of the belief that in order to, in fact, I have projects here that I'm trying to get underway right now. We need to be, so let me just say, Rytelo, the drug that I invented that just got approval for FDA, that's a telomerase inhibitor. The problem with it, and I said this 30 years ago when I first invented it, the problem is that when you inhibit telomerase, you cause the telomeres to get even more critically short, which causes more skyrocketing mutation rates, which causes cancers to come back every single time. And that's what's happening. And so Rytelo got still approved because of the fact that even though the cancers all come back, they add months to a person's life. What I'm trying to do is I'm trying to find ways of keeping telomeres long in cancer cells, actually by lengthening the telomeres in the cancer cells, and then taking the telomerase inducer away, and then treating the body with a poison that kills any cell containing telomerase. And that without causing telomere shortening. I call these, there's two projects. One is called toxic HTR. The other one's called toxic nucleotides. And I've got those projects underway here because I believe that if we could kill any cell in the body that's producing telomerase, and without letting telomeres get short, that will kill the cancers and they'll never come back. And but yeah, watch my videos, especially the one it's called, well, I think I've got it labeled as telomerase does not cause cancer, but it's from Tokyo 2017, where I've gone into great excruciating detail to explain that telomerase doesn't cause cancer, and actually telomerase decreases the risk of cancer. So sorry I spent more time than I should on that one, but it's a very important subject to me.
Okay.
What's the next question?
Dr. Nario, you got any more?
Yeah, so this one just again, I think you kind of mentioned this in passing, and you already mentioned products about this. So there is what we call mistakes or overhype approaches in anti-aging. Again, as you mentioned about lengthening telomeres and how you explained that what we should ask for in relation to quality products. So what are these other products to watch out for in terms of the claim to fame is, again, lengthening telomeres? I think he brought up in this question, they brought up some examples such as NAD boosters, senolytics. On your hit list, what are the ones that we should keep an eye out for? You kind of mentioned some a while ago.
Well, I would do NAD boosters. Okay, I think that's really good. We probably won't be necessary after we come up with something, a telomerase inducer that wins the tug of war. But taking nicotinamide riboside is, in my opinion, and I've done lots of studies, is the best way to be inducing NAD levels. The senolytics, problem with senolytics is if they're really, really successful, that can be a real problem because 90% of the cells in like a person's skin when you're 90 years old are senolytic. You don't want to be killing all those cells. Okay, but senolytics, I think, is going to be enhanced when we actually start lengthening telomeres. And we might be, as I mentioned before, already lengthening telomeres in senolytic cells because when telomeres get critically short, they become easier to lengthen. But I'm actually, I won't take any senolytics. I'm afraid of killing those cells, but when you kill them, other cells have to divide to replace those killed cells. Otherwise, you'll have like holes, microscopic holes, but they can be big holes. You need to replace those cells by cell division, and by definition, the cell division is going to cause telomere shortening. So I'm actually right now very anti-senolytics, but I would I'd be very pro-senolytics when we have telomerase inducers, unless we see what we see in vitro here in Sierra Sciences. When you induce telomerase in senolytic cells, they become unsenolytic. They're not senescent anymore. They come out of senescence. We were the first people in the world ever to prevent and reverse senescence. And a lot of people get senescence mixed up with, what's the other one? I'm going blank. I'm probably getting too tired. There's another type of, oh God, quiescence. So quiescence and senescence are very similar, but they're different mechanisms of action. And you can reverse quiescence. You just can't reverse senescence. All right.
Well, again, great
But
presentation, Dr. Bill. Thank you so
did you,
much.
did you want me to list products that don't work? And I'll tell you, I used to do that, and I got into a lot of trouble for doing that. And my lawyer has told me to quit doing it.
Well, just send it to our
Well,
private group.
Okay.
So
I can
send it to
turn,
one, don't put your name
turn-
on it.
I can turn the
And
video off, and then you can then we don't have any record of it.
Yeah.
or send us an email, and then we'll just remove your name, and we'll
Yeah,
spread it
we'll
to
just
to the
right
crowd.
Hey, was this presentation recorded?
up until now. Yeah,
Oh, good, because I thought we covered a lot of good stuff here.
we
I'd
did.
love
You
to get a
did.
record
You did
copy.
good. You know, again, like I told you at the conference, if you keep this up, we might invite you back again. You know.
Oh, I can't wait.
Yeah.
Oh, you
I've
got
been
you
loving
got
the
baited
conversation.
in.
Okay, you said the
Michael
magic word.
Oh,
Yeah.
How we doing about how about April 14th? How's that sound?
Sounds good to me.
Okay, you're on. I'll remind you.
Okay, but I'll have to look at my calendar, which I can't do right now at the moment.
All right. Well, I'll remind you.
I did
the
notice
bear
that
trap.
Sunny Win put that link into the Tokyo conference on this chat site. So if you want to get to that without having to go to my website, it's right there.
Okay, we got it. Okay, you got any more there, Dr. Nario, or are we done?
Oh, good. You got it all at the ballpark.
Okay.
I want to ask one more question. Do you know anything about redox ASEA water? They said that ASEA open up many pathway to turn on some genes, and that opens up detoxification. And some people took ASEA water and in another group, and they reverse aging 12 years.
The famous redox product.
Yeah.
It's just salt water. I mean, the recorder's
Yeah.
off, right?
Yeah.
It's like, I mean, I don't know